All right. Hello, everyone. Very happy to have the whole of you here right now for this 20th edition of the Global Community Call. I cannot believe it's the 20th edition, actually. It feels like 20 is a big number. And so yeah, and the COVID crisis is not ended at all. So we're probably actually having a 40th edition at some point in the future. We'll see what will happen. Will have happened by this time. So it's a summer edition. It's going to be shorter than usual, as everybody can understand. But we have some special guests today. Beyond just the traditional updates from the projects. There is a special project going on, started at Penn University, that is going to study the JOGL community, how how ethical guidelines emerges and are being used by the community. And so I'm very excited about that. Otherwise, we're in a moment where we're still defining the future of this program. Our main attention here is on pursuing the same approach, where we find more partners so that we can fund more projects, and we can help also find key partners to existing projects so that they can have like real impact. And in the recent weeks, I was truly happy to see that the APHP, it's a large cluster of hospitals, actually the largest cluster of hospitals in Europe, based in Paris, has accepted to test and to study some of the diagnosis tests being developed by this community. So it's very exciting, so we'll be hearing. I believe that Ali is sending some samples, some kits this week. So we should be able to have some results next week. So that's exciting. And some new developments, like we have the X Prize coming up regarding also the diagnosis test, and so it's the committee is in turmoil, trying to figure out how to participate and take part in it. I mean, having an open community initiative, and projects, being able to win such a massive prize will be quite unprecedented. So I think we have to try seriously. We have our chances. So in terms of general news and general happiness, that's probably like before the end of August, the last time I will be able to host this global community, because yes, I'm finally going to get some holidays. I cannot wait for it, as you can see from my hair. And it's going to be interesting. August is very calm period in Paris. It's almost strange when you add this to the whole of this the COVID period too. And Kemi is with me here at the JOGL office, so I'm very lucky to not be alone here. All right, so we won't have the usual update from Mark and Leo because they are on holidays too. But we'll be able to go straight to the explanation and the introduction by Alex of this Penn University project, or maybe someone else who would like to explain what you would like to do.
Well, I know a lot of you know me here. I'm Alex, but I definitely want to introduce Anna, who is the PI of this project, and Rebecca, who is our research coordinator, and Sarah and Todd are also here. They're consultants to the project. But Anna is really the leading force, so she is going to explain the project a little bit about what we're up to and what we're going to be doing.
Awesome.
Sure. Do you have the slide, Alex, or is that?
Yeah, the slide is actually linked in a comment on the, yeah, and I will add it into the, here, I'll add it into the chat so everyone can see us.
Awesome.
That's just the research team. So I will say there's 3 people who aren't on the call. Lisa Rasmussen, who's a philosopher at UNCC, Joanna Kempner, who's a sociologist at Rutgers, and Christy Guarini. So Joanna is a consultant on the project, and Christy Guarini at Baylor, who's in the bioethics department at the medical school, is also involved in the project. So, so that's the team. So it's quite a large team, and we're really excited about this project. And so basically, so this is funded by the National Science Foundation through their rapid funding that they released, rapid funding call for proposals related to COVID, specifically looking at ethical issues. And so essentially we're really interested in how ethical issues sort of bubble up and arise in do-it-yourself communities, how they're negotiated and resolved, and how participants discuss them and try to come to some resolution. And so the idea is to look, well, the methods that we're using to look at this are 2 things. So one is looking at the Slack channels and just to identify, which issues are arising. And we actually have 3 main areas that we're looking at. So biosafety and harm is one of them. Validation and replication is the second one. And attribution and credit is the third issue that we're interested in seeing. So those are the issues that we're interested in seeing how people talk about them and navigate them, essentially. And so the idea is to look through the Slack channel for discussion. Maybe we're currently developing a list of keywords that will kind of direct us to those discussions, and then follow up later on in the second phase of the project with interviews with participants who have been discussing these sort of issues on the Slack channels or just throughout the course of their projects. And so I will say that just, to end with, that the main goal, right, is to develop a better understanding of how these issues are navigated, but also to give something back to the JOGL and the open science community, just in terms of, maybe there's something we can provide by actually just doing a focused study looking at these issues. Maybe there's something that people want to hear, or guidelines or principles that we can report back. And I will say we're open to ideas and yeah, definitely feedback along the way. We have a page, project page. So my email should be up there. Lisa's is up there, Alex and Rebecca. So we're definitely open to questions and any feedback at any stage along the way.
Do you want to share a link to the project on JOGL?
Yeah. Oh. Yep, it is. It's here, I'll drop it in the chat right here.
And for how long are you going to study the?
So first, and I should talk about our opt-out. So we have opt-out mechanisms for anybody whose conversations don't want to be identified. So the grant is for 1 year. We're planning to do searches through the Slack channels for about 6 months, I would say. So looking at 6 months of data, and then the next phase will just be interviews. And then hopefully 1 year is a really short time, but hopefully have some product at the end of the year. So it's a short self-contained project.
Are you studying any other DIY communities?
So my background is I study do-it-yourself medicine in general. So I've done other work looking at DIY brain stimulation communities, do-it-yourself, people who do it-yourself fecal transplants, and do-it-yourself dentistry, which is not, and so it's interesting because the members of our team are either more involved with DIY bio stuff specifically, or have done more work in that area. So I haven't done work in that space, but I have done DIY. A lot of my work focuses on DIY medicine.
Yeah, it's cool. And do you have any ideas of questions you're going to ask the team members on JOGL?
Yes, I think that's really gonna be informed by the conversations that we find happening on the Slack channel. So, so it's really that stage 2 is really reliant on the first stage, and so we've literally there's just so much bureaucracy to get through at Penn and with getting the award and getting things started. So we're basically in the stage right now where we're sort of midway through identifying the search terms that might lead us to relevant conversations. And so that's the specific stage that we're at now.
And it's not just OpenCovid, it's other projects like iGEM, right?
No, not for this project.
Yeah,
This project is just studying the OpenCovid19 Initiative.
Okay.
Yeah, Mariana, this is Todd. I mean, one of the things that was actually quite interesting to us, particularly when the JOGL platform started on the COVID-19 things, was sort of how quickly the sort of biosafety channel sort of emerged and how those conversations were being had. And one of the actually things we're sort of interested in is yes, and what the ethical issues are, but also how this sort of distributed sort of ethical safety conversation actually happens on a system like Slack specifically, right? So that actual mechanism of collaboration is interesting to us as well, just in actually how that actually operates in a very distributed scientific research endeavor that in some instances is all being done virtually through these kinds of channels. And so that's also interesting to us to sort of see what that looks like through the lens of some of these ethical and safety issues as they pop up. And in the future, like how do you what could you might build on top of that in terms of guidances or what have you to try to frame those things for future projects like this.
Yeah, it's interesting.
Sorry, Mariam. On this matter, oh sorry, you may have an echo because I'm next to Thomas. Do you have any do you have an idea of what do you expect in terms of outcome or dissemination for this work? Are you gonna I would personally be interested in having some recommendation on how to what are the best process and having a kind of an idea of if we had to replicate such an initiative, how should we do, or should we do the same, or should we do it differently?
Yeah, absolutely. That's a great question. So we have two main dissemination goals. So really one is the academic one, which we sort of need because it's an NSF grant. So, but we've set aside funding to make sure that's completely open and not behind any paywalls. The second main dissemination is essentially exactly what you said. So recommendations based on our findings for something like this, for the future, like what have we learned and what lessons can be taken away to guide communities like this in the future. So that's our two main.
I'll be interested in the two. So
yeah, so that's our two main dissemination plans.
Right. Just want to let you know that many people here have access to the whole list of Slack channels, meeting minutes, every processes that have been set. So don't hesitate to reach out to us. You can reach out to me and or Adix. So one of us can direct you. And I know Rachel and Sarah was very involved in this. So, but if you need just the list of the Slack channels of the, you can reach out. We'll be happy to support your work.
Thank you so much. I really very much appreciate that. And so I actually should make a really big point of saying this is opt out, right? So people, if anybody's listening to this recording, we are going to be tracking everybody. If anybody does not want their messages read or analyzed or does not want to be contacted, you can opt out. Alex, maybe is the best way to email you. So we have an opt out on our project page. You can also email Alex. Alex and Rebecca are on Slack. You can contact them. And we will be posting specific opt-out messages, I believe, periodically. Is that and Alex?
If I could just mention, I had earlier on was, can we read through private messages on Slack? And we cannot do that, just so everybody knows.
Yeah, maybe we can announce your project on the announcement channel and encourage people. Maybe if you have like a consentment form, or well, maybe you probably gonna do that, or just like let them know that they can just inform us that they don't want to be ask questions. We can do that in the announcement, but also maybe on via email, maybe as well. I don't know. Yeah, that's one of the options.
Great.
Yeah, we had been, and thanks Camille for all of the support. I mean, we're so glad, and obviously, you know, we definitely, you know, love being welcomed by the community, and we had planned to post in both channels. I don't know if Billboard is still being used. It seemed like it was maybe dormant the past few weeks, but we actually do have some copy and the link that we were going to post, just sort of saying like, we're here, we're doing this, and give people a chance to opt out over the course of the next couple weeks, just in case people are traveling or whatever, not as engaged. And we want to make sure that anyone who feels like they want to has the opportunity to do so. So
yeah, that's really important.
Oh yeah, just to answer briefly, Billboard is just where we put the important documents, so it definitely makes sense that you put something there,
Sure.
and we can add up with the other. We
Yeah,
can chat
maybe
about this, the logistics.
we could post an announcement on the JOGL platform itself, a post. And there are a lot of people posting messages there. It's not just Slack. So you guys could analyze it and go to the projects page. There are some projects that are not on Slack, and there are conversations going on JOGL.
Right. Anna, is it cool if they put that slide on the on social media?
Sure. And again, as I said in the beginning, you know, if you have ideas about something you're interested in that you want us to analyze, or an ethics conversation you want to point us to that maybe we may have missed, feel free to reach out. And on the dissemination side, if you have any specific things like you think 2 months down the line, oh, this is something that would be great if that research team maybe could provide us with X or Y, you know, we're reachable and we're here. So just, you know, always feel free to contact us.
Can I just say something again? Yeah, I know this is not happening on JOGL, but I don't know if you guys would be interested in studying the people who are doing the DIY vaccines. There are scientists who are injecting themselves with COVID-19 vaccines. I guess Josiah Zahner did a live stream, or he was supposed to do a live stream yesterday with his DIY COVID vaccine. So there are a lot of biohackers doing these things now. So it would be interesting to
Yeah, the injection is on Sunday, I guess. He didn't. Yesterday was the preparation for the injection, which is happening on Sunday.
Yeah, I guess George Church did with the nasal vaccine.
Yeah. Yes. So I'm actually writing a piece for Biodesign magazine about vaccines and DIY vaccines and the concept of body autonomy. So totally separate from this project, unless someone comes into JOGL and you know, proposes to self-inject a vaccine in this community. I don't know if I mean, you know, I think we're probably pretty open, but you know, we're not focusing heavily on those projects right now.
It is very interesting, and we are.
Yeah, it is very interesting.
everybody who's on the project there's been a flurry of emails just amongst the consultants on the project and everyone who's working on it. So yeah, it is. We're following it with great interest.
Yeah, it seems like there's 3 groups now, right? It seems like there's 3 groups of DIY vaccine.
I guess you know Antonio Regalado from MIT Tech Review. He's been tweeting about this topic. He's a journalist covering this topic.
Yeah, I work with him occasionally as a journalist at Tech Review, and I have published with him before. And he's my go-to as well for new information about these kinds of situations. So for sure, he's a valuable resource to the community, I would say, for sure.
Yeah, and let us know if you have any suggestions for social media, if you have any needs or any suggestions, we can help to amplify the message.
That's awesome.
Thank you.
Thank you. Yeah, thank you.
Are we talking about the Rapid Deployment Vaccine Collaborative team?
That was the one that was the MIT group. All those people from Boston, including George Church, would be breathing it in.
Okay, because actually they just wrote to us an email saying that they would love to be able to have JOGL support. Just reading the email right now. So
I got a mail last week also from Aquarium.
That's interesting. I spoke to them and they said they had reached out to over 100 community labs. So I would assume that, that some of the folks involved with JOGL will inevitably end up crossing paths with them.
Yeah.
Yeah, it's an interesting issue. Like, do we host such an activity on JOGL or not?
When I got the email, I wrote to our Aquarium committee and sort of said, I'm not going to write back to them until we talk about it first. So we're really into the sort of do it together thing, right? But, you know, the whole risk about enhancing the disease is really
this is where we need
playing around with, I'd say.
A lot of ethical conversation.
I was gonna say.
Exactly.
Yeah, there's a need for a conversation, but yeah, a serious one.
Yeah, I wonder if in the biosafety and biosecurity guidelines that were redacted, injection or harm that could be done on oneself was covered.
I don't know if there's a specific phrase about it in our guidelines. Yeah, I would have to look at that again. It was like March or April we were putting all those things together.
Yeah, it would be interesting to have a more open discussion because, I mean, at the end, what matters is it's not about the projects, about the framework in which it's being done, right? So, clearly, you don't want to have random people injecting stuff in themselves. But if it's done with scientific expertise and guidance and control, I mean, what's the difference with another biotech startup, right?
If that it's open is better, as long as it's really well documented and so on. But I'm not sure how well it will be because, like Sarah pointed out, there's this Dave Lichina and I think this guy Justin, who we also have in our Slack group. I do think there are people who have, at least 3 different vaccines have already been taken by people that are outside of the agreed upon things. we'll have to see.
All right.
Oh, it's very funny because but the first program that JOGL launched was about vaccines, vaccines access and vaccine hesitancy, and talking with a lot of experts at international and national level. It was really a concern for everybody, because vaccines are a very different medicine, and both in terms of vaccine hesitancy but also like performing trial like random just control trials to prove the efficiency is I don't know. It's a broad topic, and it's making me a bit anxious. Like, but yeah, there's a lot of conversation that needs to happen around it. And I don't know if we can decide anything. Just I don't feel any competence of welcoming or not, or having any right to say what they should be doing.
Mm-hmm.
Well, there's a really interesting cell reports paper that I was looking at just before this call, and it looks like a lot of people do have sort of cross-reacting antibodies to SARS-CoV-2 from other coronaviruses. And this new cell reports paper really shows that there are actually antibodies that stick onto the spike protein, but they're not neutralizing. And so people could have some sort of antibody response to these various vaccines people are trying out, but it still might not protect from the virus. I mean, you really only know until you do the good study and things are controlled. When people are just injecting themselves, what's your control? I mean,
I have to run, unfortunately. I'm on childcare, but I just wanted to say thank you so much. And I really look forward to, working with you and working on this project. So.
Thank you, Anna.
Yes.
Yeah, thank you.
All right. Is there any other questions regarding this project? So good. So, do you want to introduce anything, Camille or Alex? Yeah, it's a mix of virtual and physical. Like.
I just want to mention, because we haven't announced it on the announcement channel yet, that the Corona Detective Project, the JOGL Lab, will not happen tomorrow, but next week, and we hope to have a much greater audience. So as soon as the announcement is done, or you can already communicate on it, if you want to share it with your network, join us. For 1 hour. It's next Wednesday. I'm going to write it in the chat and send you the Eventbrite link. So it's next Wednesday, 12 at 6 p.m. CET. Is it? Yeah, this same time. Next Wednesday. So instead of the global community call, we will have the JOGL Live, and Rachel will be here, probably Sarah as well. Looking forward to it.
Awesome. Do you want to provide any general updates regarding your own projects that we can share on social medias through this video? I know there has been some quite substantial improvements in the diagnosis test. Would you like to share some news?
You mean for the multiplexing? I mean, that's already old data, I feel like. But yeah, we did get a nice multiplex result that's encouraging for the Corona Detective. And yeah, we'll be talking about everything next week. And I hope also I'll have Guy and Ellen and Sarah and whoever else is involved with us. And I guess my big news of this week was that another viral target looks like it's working really well. And I don't know how well it will be in the in the context of the multiplex yet.
Hmm.
It would mean I have to order more of those expensive tagged primers, Thomas. The aquarium budget has been exhausted again.
We should open a new round of grants.
But yeah, there's definitely a lot more interesting stuff going on.
That's awesome.
Yeah, I've held off ordering my primers just to find out what washes out to be the best set, just for testing.
Well, the mammoth.
Pardon?
I was just going to say the mammoth one with the RNase P looked really good for our multiplex.
I wanted to ask Scott, how was the Reclone Symposium? Was it recorded as well?
Yes, I believe it was recorded. I had a call right at the beginning, so I came in late, and I didn't join in any of the split off groups because the tool that they used for it wasn't easy to figure out how to get into the group. But the final talk was actually quite interesting, and I think they're recording. They're already posted on the Reclone YouTube channel.
Okay, cool. I'll take a look.
Yeah.
What about you, Sarah?
So yeah, we're working. We have definitely this working test. We're still working on, so that's with the 23andMe kit that somebody could mail back, and we're still working on the point of care test. That, I guess when I say we, it's probably only me, but I'm really trying to get that working as well. So we have the kit version, the mail-in kit version is working really nicely with colorimetric lamp. But I just started trying last week the quasar lamp with the point of care stuff. So the problem is why it doesn't translate for both ways is that the buffer that comes in the kit that you would get at home and then send back is not available by itself. It's only available in the kit. And so for point of care, like at universities or factories, places like that, it's a waste to buy that collection kit. It's like $10 a kit. You don't need that at point of care. And so because they will not sell the buffer on its own, we're having to try different buffers, and that's a little bit problematic with the pH and the colorimetric lamp. So so that's why I'm now trying some fluorescent primers as well, that our collaborator at the CDC, who needs to remain anonymous, but probably most of us know who he is on this call, but he has more resources, and so he's been kind of testing those first, and then like Scott was saying, and then when they're good ones, then I'm trying them as well. So I'm hoping for some good point of care results this week using fluorescent lamp with the quasar lamp. So.
Awesome, exciting. Do we have anybody else who would like to share something? Otherwise, we'll call it done, something like that. I don't know what's the expression. I was like, I'm sure there is an expression for that. We'll call it, I don't know, something.
It's a wrap. It's a wrap.
It's a wrap. Yes.
Day. Call it a day.
We call it today. Yeah, I think that was the one. Yeah.
It's my morning.
Yeah. We call it morning. Yes.
It's even better to call it a day when it's just morning.
Yeah, same. Scott, let's just go get another cup of coffee and call it a day.
Yeah.
Thank you so much, everyone, for sharing those news with us. Thank you again. We are very good to have also the Penn University working with us on this matter. We're going to learn a lot, yeah, so the next community call will be in 2 weeks, I believe.
Yeah.
So
next week will be JOGL Live, and the following one will be Global Community.
So on the 22nd of August. And yeah, don't miss the JOGL Live with the Corona Detective Project with some awesome news and awesome people. So looking forward to seeing you all very soon. Happy summer, happy holidays for the one who will be actually enjoying it, and talk to you all very soon.
Do you want to?
Thank you.
Oh,
Thanks for having us.
Yeah. Let's do the group picture. Yeah. Let's do it. All right. Alex, Todd, would you like to activate your video stream? Also, Luba?
I would if I could, but of course our university internet has crashed because all of the students have come back.
Oh, interesting.
Welcome to university life in the new COVID outbreak.
All right. Luba, would you like to, are you there?
She's in the room next door.
Okay.
Alex, your baby is amazing.
Yeah. Yay.
Wow, so cute.
Yeah, multitasking. I heard somebody else on the call saying that they're on childcare duty.
Yeah.
Hello. This is not very COVID compliant.
All right. Hey, Miley. Thank you, everyone. Have a good one. See you next time.
Bye. Thanks, everyone.
Thank you.
Bye.
Bye.